This is Microbiology Lecture 11. All right, so we're going to talk about control of microbial growth, how it is reduced or prevented. I remind you that microbial growth refers to an increase in the number of organisms, not the size of the organisms, but the number, and that growth is typically measured exponentially or on an exponential or logarithmic scale. Okay, this is lecture 11. Now, the study of how to control microbial growth really made advances in the mid-1800s, which was just partially before and at the same time as the germ theory put forward by several scientists, including Louis Pasteur, that there were tiny organisms that could account for that grew and infected people and could account for disease. Just at the same time, and in one case not knowing, not having yet seen any of those results, Ignaz Semmelweis put forward the idea that patients would do much better if medical staff cleaned their hands properly. And then later, after Pasteur had put forward his germ theory, Joseph Lister also did the same thing separately in England. Ignaz Semmelweis was in Hungary. Semmelweis was a Hungarian doctor who saw that the patients of those who had just performed autopsies had a much higher frequency of infection. Patients, and we're talking about patients, I left it out here, patients, obstetric patients had a much lower frequency of infection than the patients of midwives who did not perform autopsies. So he proposed that an infection was spread by the contaminated, and I should point out unwashed, hands of physicians or medical students. He recommended that chlorine should be used before attending to a physician. His findings were ignored, dismissed, ridiculed, even though his mortality rate, he showed a mortality rate drop from 18% to approximately 2% or less. In fact, he was dismissed from the university soon after that, actually, not too long after that. His proposal and his findings and his results were the first strong evidence of the benefit of aseptic technique, removing pathogens to the benefit of the patient and reduction towards reducing infectious disease. It was, again, as I said, proposed before the development of the germ theory. Well, Semmelweis, this is some data relating to his findings. It was from Vienna, where they took up his findings in 1847 and showed a tremendous drop in mortality rate from puerperal fever and infections seen in obstetric patients. I should add that in 1969, more than 100 years after, the university from which he was dismissed was renamed Semmelweis University. Lister, another giant name in development of aseptic techniques, he applied the germ theory to medical procedures. The idea that there were microscopic organisms that could multiply and cause disease, and that they had to be removed, and procedures had to be done either using aseptic technique or sterile technique for surgery. He began soaking his surgical dressings in a mild solution, a phenol solution, a dilute solution of phenol. Phenol can be fairly corrosive even. And he showed a drastic reduction in incidence of infection and death. One major difference was he was able to sell his ideas using more persuasive approaches, and he had the evidence, as did Semmelweis, but he was more successful at convincing his colleagues, and his practice was quickly adopted and soon became the standard and widespread. So, in terms of control of the terminology associated with control microbial growth that you need to know, and I'll use in this lecture, is sepsis is contamination by microbes. Asepsis is the absence of significant contamination. It does not mean sterile. It means free of pathogens. So aseptic techniques prevent microbial contamination with pathogens. So you're removing pathogens. It does not mean that you're working in a 100% sterile environment, which is quite difficult to do. Antiseptic technique, is removal of pathogens from living tissue. There is a subtle but very important difference between sterility and aseptic technique, sterile technique and aseptic technique. Sterile technique, it does not happen in a surgical theater where there's people standing in it. There's going to always be some bacteria, but the room is treated so that it's aseptically prepared for the next surgery. It's cleaned up as best as possible using fairly thorough techniques, but it's not sterile. Sterile means absolutely nothing. That's not that easy to achieve. Sterilization means removal of all microbial life. There's more terms you need to know. Disinfection is, again, the same idea as antisepsis, removal of pathogens. You hear about sanitizations, and you might see it on, that your utensils that you're using have been sanitized. It does not mean sterile. It means that they've reduced the microbial count. Okay, it's just a reduction, and there's a lower microbial count. There's still microbes there. A germicide or a biocide is something that kills microbial organisms. Degerming is removal of most microbes, not all, from a very limited area. And that's what happens when you swab an area prior to injection. You're degerming. So you're removing most, not all, of the microbial organisms from that area of the skin that you're about to inject into. This is a term I doubt I'll use again, but stasis, bacteriostasis, stasis refers to inhibition of growth as opposed to killing of organisms. Bactericidal, microbicidal, germicidal, all those refer to killing, but stasis, if the word stasis is tacked on, it's not killing but inhibition of growth. Okay, just as bacterial populations grow at an exponential rate, they also die at an exponential rate. And what you see here is, although it's so this is a logarithmic scale of number of surviving microbial organisms. Logarithmic scale of number of microbial survivors, I guess, surviving the treatment. And these are arithmetic numbers of microbial survivors. So it refers to these numbers on this scale here on this side, and this refers to the numbers here on this side. This logarithmic number of surviving organisms refers to this scale. So you can see rapid decline with treatment. Okay, and one key factor is time in the treatment, how long the timing is for the treatment. There's many ways of cleaning surfaces or cleaning hands, but the time that the agent you're using is on that surface or on the hands is extremely important because the effect increases with time. There are many factors that are important, but one key one is time, and that's why it's shown here in this diagram. So among the factors which I already mentioned one was time of exposure to an antimicrobial treatment. The other key factors are the number of microbes or the load, the initial number of microbes or the load. If you start off with a high initial number, then it will be a logarithmic scale type of progression of death, and the load and the number of organisms surviving with time drops, and it is at the same rate as if you start at the lower population number, but you reach down to zero or close to zero levels much faster because you start with a much smaller population. So time of exposure is important, shown here, and as it progresses down with time, but also the number initial load is very important. And then there are, of course, the characteristics of the microbes themselves, the environment in which they are present. If there's a lot of organic matter, it may misdirect the action of the, whatever you're trying to sterile or treat with, and it may soak it up or reduce the effectiveness. So there's the environmental circumstances, how much other organic matter there is, whether it's within a biofilm or not. I'll talk about biofilms later. Okay. The action of control agents, any agent that can have an effect on microorganisms, the actions can be grouped into several large groups. Some alter the membrane permeability, the permeability of the bacterial plasma membrane or cell membrane. Others damage proteins, and some damage nucleic acids. There are two major methods of approach to control of microbial growth. One group of methods are physical, and the other are chemical. And we're going to go through the physical ones first, and then the chemical ones. Many of these you use in day-to-day life, just preparing food, cleaning yourself, things like that. I mean, the idea is to not have microorganisms present with your food and not have excessive potential pathogenic organisms associated with yourself, right? Okay, so some of these are very much common sense when you think about it, and some, even though they're chemicals and you haven't heard the chemical terms, you're actually quite familiar with the kind of products that contain the chemicals that I'm going to describe. Let's start with the first physical method is heat. For every organism there is a thermal death point, TDP, a thermal death point, the lowest temperature at which all the cells in a liquid culture can be killed after being treated for 10 minutes at that temperature. Thermal death time is the time to kill that it takes to kill all of the cells, okay, in a liquid culture. So at a particular temperature, given temperature, what's the thermal death time? You get it, if it takes 2 minutes or 10 minutes or half an hour of treatment at that time, then they have different thermal death times. Thermal death point, that's the lowest temperature at which everything gets killed within 10 minutes. So, will 60 degrees centigrade kill everything in 10 minutes? Will 100 degrees centigrade kill everything in 10 minutes? There's different thermal death points. So in terms of temperature, there are several different ways of approaching it. So we're talking about heat as a physical method for control of microbial growth. One way is using moist heat. And the thing about moist heat is, first of all, the heat will denature proteins. And as you know, denaturation means an unfolding, change in shape of the proteins. Basically, a lot of the, certainly the hydrogen bonding comes apart. Sometimes even covalent bonds can come apart. To kill all organisms, usually 10 to 20 minutes of boiling temperature is adequate. However, some bacteria that can produce endospores can resist being killed completely even after 20 hours of boiling. And so what is added is pressure. Autoclave steam under pressure is much more effective. At a pressure of 15 pounds per square inch, and the steam will be at a higher temperature at these higher pressures. The higher the pressure, the more it takes to get the water into a gas form, steam, and the water will be at the steam will be at 121 degrees. Everything is killed, including spores, after 15 minutes. So these are the conditions in an autoclave: 15 pounds per square inch, 121 degrees Celsius, the steam inside, and 15 minutes is enough to kill everything. And usually things are autoclaved 20 to 30 minutes, just to make sure. It's a chamber that you put things into and close a big steel door with a wheel on it. You spin, and it pipes in at a high pressure air and also steam and steam into the chamber, and it heats it up and pressurizes the chamber to 15 pounds per square inch, and the water vapor, gaseous water is 121 degrees Celsius. The relationship between pressure and temperature of the steam is, as I said, the temperature at which the water will boil goes up with increasing pressure. Here you see and this is relative to how much more than standard atmospheric pressure, okay? So standard atmospheric pressure at sea level is 1 atmosphere, and so if you're 0 above 1 atmosphere, then water will boil at 100 degrees. If you're 5 pounds per square inch above sea level atmosphere, then water boils at 110. And as I said before, in an autoclave, the pressure will be 15 pounds per square inch, so water will be boiling at 121 degrees. The opposite occurs if you are at a lower pressure. So the higher in elevation you go, the lower the boiling point, and in fact at a very high elevation it's difficult to boil water. You can boil it it's difficult to get a hot enough water to make tea or coffee. It'll boil, but it won't be particularly hot. Okay, that was moist heat. Another type of heat application to control microbial growth is pasteurization, named after Louis Pasteur, who was the main proponent to put forward the germ theory of microbial life forms. In other words, tiny microscopic living organisms that can contaminate and grow and infect and cause disease. Pasteurization is a procedure that involves using warm water, hot water, 63 degrees Celsius for 30 minutes. Now, pasteurization reduces organisms and the number of organisms that could spoil food, reduces the number of pathogens, but it is not a sterilization. In order to sterilize, you have to boil for quite a while, or better still, put in an autoclave. So you're at high pressure, higher temperature of steam. It is a mild kind of heating. You can stick your hand in 63 degrees water. You're not going to burn yourself. All of the procedures below are equivalent in terms of their microbicidal ability. You know, we already talked about the temperature versus time application. 63 for 30 minutes, which is pasteurization, has the same effect as 72 for 15 seconds, or 140 for 1 second. And, you know, we do treat certain things by pasteurization, and other processes, other kinds of foods are treated. This is mostly applied to food, to reduce things that spoil that spoil the food, reduce the number of organisms present that could spoil the food. A lot of the original research done in the 1800s, mid 1800s, was in fact sponsored by the food industry, cheese, wine, beer manufacturers, who often had spoiled batches because not that they realized it at the time, but other organisms were getting into their food during the preparation manufacturing phase and was spoiling it. So they were paying people to do research on that, and that's where a lot of their original funding came from that led to the development of the germ theory and understanding of microbial organisms and their growth. Basically, there are always organisms that can survive pasteurization. These are called thermoduric organisms. They thermo from heat, duric from resistance or hardness. They can tolerate heat and survive. You know, pasteurization is used for quite a few different preparation of quite a few different foodstuffs, particularly milk. But even if the milk inside the milk carton, the whole thing was pasteurized, if you keep it long enough, there will be organisms in there. There will be some organisms in there that eventually grow. It takes a long time because there's such small numbers, but they'll eventually take off. You can't keep it forever and expect it not to spoil. It will spoil because these reduce. They do not. They reduce, not sterilize. This reduces the number of organisms. So we did moist heat pasteurization, dry heat sterilization, a different approach. This kills by process of oxidation, flaming, incineration, hot air sterilization. Flaming your loop, it's a method of sterilizing, right? You want to use a sterile loop to take a sample because you don't want the other things in your sample when you're going to inoculate something. Hot air, you know, there is a big difference in moist, high pressure in an autoclave that is effective at even killing endospores, and hot air. If it's dry, then it has to be at a higher temperature for a longer period of time before it is effective at killing things. These, of course, are much higher than 170 degrees Celsius. Okay, we covered heat. Now, a second method of physical control is through filtration, which involves actually passing either a gas, air, or liquid through a filter, and the filter removes all the microbes. And these kind of highly effective, very small pore filters that can remove microorganisms are called HEPA filters. And they're the kind of things that are used for the air circulating in surgical theaters or in containment hoods where you have to do sterile work. Or actually now they're becoming popular for air filtration in houses, although it's a little ridiculous to have a HEPA filter in a house. I mean, you can't stop. There's so much more coming in from outside. But another way of physical method of controlling microbial growth is by low temperature. which is used in the food industry. Flash freezing of fish will kill many parasitic worms. It certainly inhibits all organisms' growth, including microorganisms, and you use this daily in your own home to reduce growth in food. You preserve food by refrigerating. The same can be done for materials that are used with patients, injected into patients, that are refrigerated or deep frozen. Very effective way of preservation is lyophilization, which is freeze drying. It removes all water. Freeze drying will remove all the water from substance, and if there's absolutely no water, then there will be no growth of any bacterial organism. Another method is high pressure. It is a way of denaturing proteins, and in that way, in the microorganism, in that way, preventing them from being able to grow. It has been used in the food industry for fruit juices. Drying or desiccation prevents any metabolic activity for microorganisms and therefore any microbial growth, and has been used for fish preparation, preservation. Osmotic pressure is another way. This involves use of salt. A lot of these are for food, physical methods in food production preservation, but many are also applied, not these of course, salting, but many are used for preservation of pharmacological agents. Salting, what that does is it pulls water by osmosis out of the cells and causes plasmolysis and stops any growth. Okay, the last one that we're going to discuss is the physical method, is using radiation, which has a strong enough energy to actually damage the DNA and prevent the organism from multiplying. Ionizing radiation includes X-rays, gamma rays, electron beam. All of these can damage the DNA sufficiently to prevent DNA replication occurring. There are also non-ionizing radiation methods like UV, ultraviolet radiation, very effective in preventing or killing off microorganisms. It's used in water supplies. It's used very effectively for foods that are laid out in the sun. Not only get dried, but also organisms are killed by ultraviolet rays from the sun. I should point out that microwaves, in other words, the microwave in the oven, microwave oven, have no such antimicrobial effect. Okay, this is a spectrum looking at the wavelength versus energy. The shorter the wavelength, the higher the energy. So these are short wavelength, high energy down at this end, at the very low end, and that's where you get all of the bactericidal effects. Let's go to chemical methods of control of microbial growth. Essentially, this is disinfection. Very few of these provide or cause sterility. These are not methods that are used for sterilization, but they significantly reduce the number of microorganisms. They are, however, and you always have to keep this in mind, they are selectively effective. They are effective more against one type of organism than another, depending upon which chemical compound you use. So some are very effective with certain types, and others are effective against other types of microorganisms. The main principles behind disinfection are that there are a number of factors influencing their effectiveness. Much like heat, I discussed different aspects of the effectiveness of heat. Here we're talking about chemical methods, and you have to take several factors into mind. The concentration of your disinfectant, how concentrated it is in the preparation you're using, the time involved, how long you treat it, the pH, and how much other organic matter is present that could interfere with the effect of the chemical compound on the microorganisms themselves. We'll take these one at a time. So one thing you should be aware of is in order to evaluate the effectiveness of a disinfectant agent, quite often there are tests that are done. One example is called the use dilution test, where you dip metal rings in bacteria. They're dried, so the bacteria dry onto them, and then they're placed in the disinfectant for a fixed amount of time at a particular temperature, and then they're transferred to culture to see whether the bacteria survive or not. So the rings are placed on nutrient agar plates and petri dishes, and or into broth cultures to see how well the disinfectant worked. Disc diffusion is widely used, and you'll do that in the lab. Disc diffusion involves soaking the agent into a round piece of filter paper and then placing them on a petri dish. Immediately after the petri dish is streaked in such a way that you'll get what's called a bacterial lawn. So it's streaked completely covered with bacteria using a bent glass rod, and the bacteria are inoculated over the entire surface. So 24 hours later there'll be a solid growth. All the colonies will be confluent with each other, touching each other. So it'll be a solid growth. On top of that plate, you place the filter discs containing different chemical agents that you're trying to test. And what you're looking for is, and then their plates put in an incubator. And essentially what you're looking for is the results shown down below. Is there effective inhibition of growth in the area surrounding the filter paper, where the chemical compound diffused away from the disc? Sometimes you get extremely large, much larger than shown here, areas. These areas are called the zone of inhibition. Let's see if I can, zone of inhibition. So if you see a large zone of inhibition, it's an effective control for that particular microorganism, and different organisms have different sensitivities to different chemical compounds. Here, for instance, you see that this phenol reagent is not effective against Pseudomonas aeruginosa, but certainly effective against these other two, Staphylococcus aureus and E. coli. Okay, what kind of disinfectant molecules are there? Well, they're large group. They're grouped into different chemical compounds. Phenols all have this kind of structure in them. That's the common thread for phenols. And phenols or derivatives of phenol include things like Lysol or Physohex, as shown here in these pictures, but they are quite active, stable. They persist for a long time. It's not as if it's only good for a few minutes. They will persist once applied for a long time. There are phenols and bisphenols, or phenolic or chemical compounds, and bisphenolic chemical compounds. Let's move on. Biguanides is the second class, and that includes chlorhexidine gluconate, which is used in strong mouthwashes. These kind of mouthwashes, by the way, they're very effective. They even are effective against endospores, but prolonged use can stain your teeth if you use it for more than just a few days. It's typically used post-surgical procedures. Halogens. The halogens include iodine and chlorine, very effective, strong oxidizing agents that you wouldn't want on your skin. I mean, this is essentially bleach. If it's chlorine, it's bleach. It's hypochlorous acid. Some can be used directly on tissues, and others cannot. They're too strong, so they can only be used on instruments or tools or things that need to be used, that need to be sterile. Alcohol is quite an interesting, commonly used disinfectant. Ethanol can be used, but usually it's isopropanol. Isopropanol is very effective, and you don't get people drinking it because it's poisonous. So they don't sell cheap ethanol because otherwise people would drink it. It is bactericidal and fungicidal. It is good against bacteria and fungi, but it is ineffective on endospores. It is extremely effective on enveloped viruses. It destroys the envelope of enveloped viruses. It's effective through its action on proteins and lipids. That's why, if it dissolves lipids, it'll dissolve the phospholipid bilayer of an envelope, right? It is not good on wounds at all. It's painful on wounds, in fact, but it is good for degerming injection sites. One interesting point, important point actually, this is data from disinfection using ethanol against Streptococcus pyogenes, or group A Streptococcus gas organisms. The important thing to note is that it's not 100% that's most effective. In fact, the most effective, generally accepted most effective dose, it's not shown in this data. Actually, the data is not a good indicator, but 70% ethanol is typically the dosage concentration that's used for disinfection. And that's the most effective way. It's the water content acts as a wetting agent and keeps the alcohol present for a little bit longer, so you effectively are increasing the time. In the end, the best kind of disinfectant is soap and warm water. The reason being is because it is the most effective at removal. It removes pathogens really well. If you wash your hands using soap and you do it properly, in other words, you cover all the surfaces, you scrub all the surfaces using soap. It doesn't have to be that hard to scrub. The proper technique for a long enough period of time, certainly 20 seconds is plenty, you will very effectively remove pathogens, and that's an excellent way of disinfecting. Heavy metals are effective and yet poisonous, so let's move on. Detergents, soaps and detergents are used. Other types of soaps and detergents. There are acid or anionic detergents, and there are cationic detergents. One commonly used one, and it's referred to. You don't see it under its full name. They're commonly referred to as quats. Are quaternary ammonium compounds, or quats. This is the kind of thing you actually find in Pine Sol or Fantastic all-purpose cleaner. They're fairly mild disinfecting agents. They're good for bactericidal activity, and virucidal means killing viruses. Bactericidal, of course, is killing bacteria. Good for enveloped viruses. Soaps, detergents are good. Let's move to chemical preservatives, as opposed to chemicals that can be used for surfaces or hands, or tissue on the body. Let's go to chemical preservatives. They include things like organic acids, are commonly used to preserve foods. Nitrite is very effective at preserving food and making meat safe to consume, even though it hasn't been refrigerated, and that's what provides the pink color in cold cuts, sausage, things like that. It's important because it's very effective at preventing endospore germination. Really does a good job of preventing bacterial growth in meat. It mimics the action of actual smoke from a fire. If you look at smoked meat that was smoked on a fire, it'll have this pink color. That pink color are the nitrites in the meat, but it also is commonly added. For sausages, it's added in, giving them that pink color. It's not a natural smoking; it's addition of nitrite. Okay, aldehydes. These cannot be used with food. I'm not sure why they're here. Oh, it was as a separate page, one page for preserving of food. Aldehydes, there's no way you can add those to foods. There are also gaseous ways of sterilizing, disinfecting. Commonly used one is ethylene oxide. It'll kill microbes and endospores. Oxidizing agents, superoxide, like agents like hydrogen peroxide, again, effective on objects, but not too powerful for tissue. In general, if you look at different agents, and we did talk about phenols and quats and halogens like chlorines and aldehydes and alcohol, they have different effectiveness against different types of microbial organisms or microbial structures. Some of the most resistant types include endospores from Clostridia, the genus Clostridium, and Bacillus, and also Mycobacterium, the genus that includes organisms that cause tuberculosis and leprosy. These are often used as examples of different effectiveness of different agents, and you can see the differences shown for many of these. I point out to you here that alcohol is very poor against endospores, but quite good for Mycobacterium. In general, if you look at it overall, and this is, you have to take this into consideration that it's not simplistic, simple graph, most resistant, least resistant. It also depends on how much time, what agent, what concentration, how much other matter is present. But in general, the most resistant, the most difficult to control are things like prions or endospores or Mycobacterium, and the easiest ones to control, and easy still can have a serious problem, are some of the easiest to destroy are viruses with lipid envelopes. Now, flu, coronavirus, COVID-19, both have lipid envelopes. Easy to destroy relative to other things like these. However, sometimes they're so omnipresent that it's difficult to avoid them. So, there's a lot of things to take into consideration. It does not mean that it's easy to kill them off, and therefore we have nothing to worry about with enveloped viruses. We do. All right, that's the end of lecture 11 for microbiology.